everyone. Welcome to GPRC Colloquium series. Today's the speaker is Changjun Justin Lee. So he is the director of Center for Cognition and Sociality in Institute for Basic Science, so-called IBS in Daejeon. He will talk about the reactive astrocytes as the perfect source of stem cells in the brain. So he is a famous researcher in the field of glial cells, and especially astrocytes are his main topic, and he's a leading scientist in the world. So I think all of us participants and online the viewers will have benefit from his talk. So please welcome Dr. Lee.
Okay, thank you very much. Thank you very much for inviting me to this pain consortium, and I'm happy that some of our research might be helpful for this pain research. Now, the title of my talk, there was some mix-up here. I recently submitted this reactive astrocyte as the cause of Alzheimer's disease, but the original title was this stem cell. Is that correct?
Yeah. Oh, so that part is gone in this real title.
I see. Okay. Do you think I should change it, or?
No, it's okay. So I think you can say anything then as you planned.
Okay. All right. Now, I believe this talk will give some overview of what astrocytes are doing in the brain, and also what goes wrong in a disease state like Alzheimer's disease. And simply put, astrocytes become reactive, and we have accumulated evidence that this reactive astrocyte is not just a byproduct of the disease states, but it can be the cause of the various diseases, including Alzheimer's disease. So I'm hoping that after this talk, your group, the pain interested people, can take some of the ideas that I introduced to you today and apply to pain field, because I have applied this idea to various diseases such as Parkinson disease, stroke, recovery after stroke, but we have not really applied this idea to pain field. So most of the tools that we have developed can be utilized for pain research. Okay, so briefly, Alzheimer's disease, as you know, is a disease affecting old people. It causes brain atrophy like this one in a severe condition. You can see that the brain shrinks. Most of the brain shows brain atrophy, especially in this region of temporal cortex and near the entorhinal cortex and hippocampus. Now, many people believe that there are many neurons are dying or dead already. That's why it causes this shrinkage and the brain atrophy. But not many people actually know that in this remaining brain of the Alzheimer's disease patient brain, there are many glial cells, and especially reactive astrocytes and reactive microglia. So not many people actually know that. So I mean, so far the cause of disease has been proposed to be this amyloid plaques that are accumulated and deposited over the time, and this causes the Alzheimer's disease. However, this amyloid hypothesis-based drugs have failed recently, as you know, and many drugs, especially the antibody-based drugs that are supposed to clear the amyloid plaques from the Alzheimer's disease patient brain have failed. Although recently aducanumab, which was approved by FDA, US FDA, but this drug still struggles. And recently I heard that one patient died from taking this aducanumab, and the price of the drug has gone down and to the point of like half price. And the future of this aducanumab is not very bright. And so these failures of continuous failures of amyloid hypothesis-based drugs calls for a desperate need for reestablishment of the hypothesis. So the proposed causes of Alzheimer's disease is basically the initial hypothesis, amyloid hypothesis, and people have believed that the amyloid plaques on the left somehow causes all these symptoms of the Alzheimer's disease, like tauopathy, neuronal death, brain atrophy, and finally the cognitive decline. However, due to these failures, there's this question mark in the middle, and people now starting to believe that there's some missing link between the amyloid plaques and all these symptoms of the Alzheimer's disease. And to make the story short, I would just tell you the bottom line answer to this. I think the missing link is this neuroinflammation that is probably triggered by amyloid plaques, but in the later on, at the late stage, this neuroinflammation becomes the dominant feature of the Alzheimer's disease, so that at the end, even if you remove the amyloid plaques, it doesn't matter anymore because the neuroinflammation continues to cause tauopathy, neuronal death, and brain atrophy and cognitive decline. So the question is, what is this neuroinflammation? Well, if you look at the history behind this Alzheimer's disease, the original person who reported this Alzheimer's disease is called Alois Alzheimer, as you see in this picture. He was a doctor, and he diagnosed a person with Alzheimer's disease, and he called the disease Alzheimer's disease. And he reported from the post-mortem brain sample of his patient. He reported actually these features of the pathological features of this brain. And if you look at the recent textbooks, you will find that Alzheimer's disease has these two features: amyloid plaques and neurofibrillary tangles. Well, Alzheimer himself reported these two features, as well as the third feature, which is this reactive astrocyte, and he called it the glia here. So he saw many gliocysts here, and many glial cells became reactive. So here's a feature of a third component that the Alois Alzheimer reported in his original report. But later on, it became, it disappeared from the textbooks, as you can see, and most of the people focused on the amyloid plaques and these tangles. This is the famous textbook of Alzheimer's disease, and you only see these two features. So what happened? I mean, why do people ignore this third feature, the reactive astrogliosis, and only focused on these two features? Well, I don't know. I mean, there's people initially thought that this reactive astrogliosis is the byproduct of the neuronal death and this amyloid plaques. So that's why people tended to just ignore it. But if you look at this time course of the disease progression, you can see that in the early stage, even before the pre-symptomatic phase, you will see that the astrocytic dysfunction occurs very early, way before the microglia activation and this neuronal dysfunction by FDG PET and the hippocampal brain shrinkage. So astrocytic dysfunction appears to be very important, but somehow disappeared from the history, and people have focused not on these glial cells, but instead of glial cells, they focus on the neuronal dysfunction. And we have focused on this reactive astrocyte and MAO-B enzyme in particular, and we decided to call ourselves a Maoist. And there's a big fight between Baptists who believe beta amyloid protein is the cause, and there are also Taoists who believe that tau protein hypophosphorylation is the cause. We believe it's reactive astrocyte, and it's through the MAO-B, so we call ourselves a Maoist. So we are the third group of the people who are actually kind of fighting each other. Okay, so reactive astrocytes appear in almost everywhere whenever there's a neuroinflammation. So any disease that accompanies neuroinflammation, you will find the reactive astrocyte. For example, in Huntington's disease, and Alzheimer's disease, and ALS, and Parkinson's disease, and multiple sclerosis. The feature of this reactive astrocyte is that they are kind of express high level of S100 beta or GFAP, astrocytic marker, and you will see the hypertrophy of the astrocytes. And this has become the classical feature of this neuroinflammation accompanying brain diseases. And this reactive astrocyte is the major component of the brain inflammation. Okay, so now we in 2014 we have published that the first report about this reactive astrocyte involved in Alzheimer's disease. We have demonstrated that reactive astrocytes produces GABA by the action of the MAO-B enzyme, monoamine oxidase B, and this GABA tonically is released from the reactive astrocyte and strongly inhibits the neighboring neurons presynaptically and postsynaptically. And as you see in this picture, you see that the astrocyte in the normal condition does not contain this GABA in the red color. However, the neighboring neuron has high level of GABA, and this is a typical GABA production by neuron, GABAergic neurons. In APP-PS1 mouse, which is a popular Alzheimer's disease model, you will see that there's hypertrophy and GFAP expression, high level of GFAP expression. And in this reactive astrocyte, you will see that the GABA suddenly appears and is as high as the neighboring neurons, as you see in this arrowhead. So, and this is near the plaque. This blue is a plaque. So near the plaque, we found many of these reactive astrocytes starting to produce GABA, and this GABA strongly inhibits the neuronal activity, and this causes Alzheimer's disease. So we have this movie demonstrating this mechanism. I'll be showing a lot of these movies instead of data. We have a lot of data, but I think it's better to show this mechanistic video. So normally a person navigates to find the way back home, and the neurons has to fire action potentials to actually achieve this navigation. But when this firing is stopped, the person can no longer find the way back home. So we found that under normal conditions, spike occurs like this, but under pathological condition, when amyloid plaques are abundant, astrocytes take up this amyloid beta, and this produces putrescine, which is polyamine produced by astrocyte. And the presence of polyamine makes the putrescine degradation pathway turn down, and this MAO-B makes the GABA, and this GABA inhibits neuron, and the firing disappears. Now, if you inhibit the MAO-B by cellular, which is MAO-B inhibitor, we were able to reverse this inhibition, and then we could almost instantly recover the animals from the memory impairment. So that was our original report in 2014. And at that time, when we were publishing this paper, the first author at that time was a student, Sunmi Jo. Now she's a PI. At that time, we designed or developed this metabolic pathway starting from the A beta oligomer, taken up through endocytosis and goes through the autophagy system, and autophagy to lysosome, autophagolysosome. And then this production or degradation of the protein leads to production of amino acids and ammonia. And then this turns on the urea cycle. And then urea cycle, and one of the product of the urea cycle is ornithine. And this ornithine leads to production of putrescine. Now the putrescine goes through, and then through MAO-B, GABA is produced. So we actually proposed a diagram, a mechanistic diagram of this entire pathway at that time. And we are systematically, one at a time, from the bottom up, we were trying to demonstrate whether this whole pathway exists. So that was 2014, about 8 years ago. Since then, we have developed this reversible MAO-B inhibitor because the irreversible MAO-B inhibitor like selagelin had a problem. So if we use selagelin for 1 or 2 weeks, the drug worked perfectly by blocking the GABA synthesis. But if we used for about 3-4 weeks, longer than 2 weeks, this prolonged use of the drug selagelin, for example, somehow reverted the GABA production. And we decided that we should make a reversible MAO-B inhibitor. So we made this KDS 2010, and it turned out that this use of this reversible MAO-B inhibitor, we were able to circumvent all these problems of the irreversible inhibitors such as selagelin, so that when we use this KDS 2010 for 4 weeks, we were able to continuously block the GABA synthesis, and at the same time, the astrogliosis was also inhibited continuously for a long time. So this was a very powerful drug that we could use as a drug that inhibits the astrogliosis or reactive astrocyte and also GABA production. So this is the video of how this MAO-B, reversible MAO-B inhibitor was developed, and the effect of this MAO-B inhibitor. So in Alzheimer's disease, there are amyloid beta plaques, which causes neuronal death. And surrounding the plaques, we see this reactive astrocyte, and it produces GABA through the enzyme. Here is the MAO-B. When A beta is taken up, astrocytes produce putrescine, and this putrescine is further degraded to cause production of GABA through the MAO-B enzyme. And this process leads to the neuronal inhibition. So we developed this KDS 2010, which is a reversible inhibitor compared to selagelin. Notice that selagelin has this triple bond here, which is permanently blocks the enzyme, whereas KDS 2010 doesn't have that. So that's why KDS 2010 serves as a reversible inhibitor. So KDS 2010 enzyme binds to the enzyme, and it also gets off and on and off as a competitive inhibitor. And this drug turns out to have excellent druggability with excellent solubility and BBB permeability. And so KDS 2000 was very useful for long-term treatment. And we were able to show that this selagelin, compared to selagelin treated KDS 2010 showed much more efficient and superior activity. So this was published in Science Advances in 2018, I think. Okay, so we are now equipped with this perfect, very nice, excellent KDS 2010. And using this drug, and also we use various genetic models, such as MAO-B shRNA, MAO-B knockout, and MAO-B overexpression mouse. We were able to test the idea of inhibiting MAO-B, and we found that the inhibition of MAO-B prevents the reactive astrogliosis and scar formation in the stab wound injury model. So when there's a stab wound, it causes a severe reactive astrocyte lining up this injury site, and also some tangential as well as radial reactive astrocytes. And these all contain the MAO-B, a high level of MAO-B, and this causes the production of GABA. At the same time, MAO-B can produce H2O2. And these two toxic molecules causes hypertrophy and scar formation. And when we inhibited this MAO-B from these cells, we were able to reduce GABA production and H2O2 production, and the hypertrophy and scar was prevented. So this was recently published, actually this year. This kind of shows the powerful nature of the drug, MAO-B inhibitor, as well as the knockout, MAO-B knockout, and shRNA that we developed. Now, using the KDS 2010, we were able to show from the bench to the bedside, hopefully. So in 2010, we firstly show the astrocytes produces GABA and release GABA through the BEST1 channel. And this, in the following paper, we were able to show GABA production through the MAO-B pathway, through the putrescine degradation pathway. And in the following paper, we show that the MAO-B it can be a therapeutic target against Alzheimer's disease. And we developed finally developed reversible MAO-B inhibitor called KDS 2010. And this drug was transferred to a company called Neurobiogen, and they just got the approval for IND, and they are currently doing the first stage of the clinical test. So we are actually going from the bench to the bedside through using this KDS 2010. Using this drug, we also collaborated with many clinicians, shown here. We have already collaborated with Professor Hyung-il Kim at GIST for the effect of these MAO-B inhibitors on the white matter stroke and the recovery after stroke. And also for Parkinson disease, we show that we collaborated with Dr. Sang Myung Chun at Asan Medical. And we published that in the Parkinson disease, a similar mechanism exists. And at Yonsei University, we are still working on the spinal cord injury and recovery after spinal cord injury. And we have also looked at the obesity and the role of this tonic GABA from reactive astrocyte and its obesity. This is actually under revision now. We also collaborated with Dr. Jung Sang-yoon at Cha Bundang Cha Hospital, and we studied the role of this reactive astrocyte and GABA in the hippocampus of the rheumatoid arthritis patients, and found that this KDS 2010 is very effective, not only improving the cognitive impairment in rheumatoid arthritis, but also rheumatoid arthritis itself. So this could be a very good drug. And so we are actually looking for some good collaborators for pain research. So this drug should be tested for pain in various pain models. Okay, so the reactive astrocytes, if I give a little bit more introduction, there are actually two types. So it's been classified that there exists a mild or moderate reactive astrocyte under kind of mild, moderate insult condition. These astrocytes from healthy condition to become like a hypertrophy. And you can see some molecular changes and functional changes. But if you wait a little bit, a long time, they actually recover to the back to the healthy condition. So these recovering reactive astrocytes are called mild reactive astrocyte. However, under severe insult conditions, these astrocytes become heavily changed in terms of the morphology, and they start to form a glial scar, and they start to form a barrier, and this barrier can actually inhibit the axon regeneration, and this scar formation is almost irreversible. So once this scar is formed, it's very difficult to reverse this process. So this irreversible scar forming severe reactive astrocyte also exists. And so far, this molecular, the morphological changes has been reported, but the molecular changes and their mechanism and their function is not very well studied yet. So in terms of Alzheimer's disease, we had this hypothesis that this reactive astrocyte must do something very critical for the disease progression. So we made this model mouse called GID mouse, which is a combination or crossing of this GFAP CRE-RT2 mouse crossed with the this ROSA 26 DTR mouse with a fluxed stop codon. So with tamoxifen induction, this mouse starts to express diphtheria toxin receptor from the astrocyte only. And if we add diphtheria toxin, the astrocyte become either mildly reactive or severely reactive, depending on the dose of the diphtheria toxin. So this was very convenient because using this model, we were able to bypass the amyloid plaques. So without amyloid plaques, we were able to demonstrate that is if we induce severe reactive astrocyte, but not mildly reactive astrocyte, but severe reactive astrocyte by adding enough diphtheria toxin, we were able to recapitulate or reproduce all the features of Alzheimer's disease. For example, tauopathy we saw. We also saw neurodegeneration. We also saw permanent cognitive impairment and brain atrophy and progressive death from the whole animal actually died. So, and all this was mediated by H2O2, which is hydrogen peroxide, which is also one of the byproducts of the MAO-B action. So MAO-B not only produces GABA, but it also produces H2O2. And this accumulated H2O2 can turn on INOS and superoxide producing NOx2, which leads to production of peroxynitrite, which is a deadly kind of atomic bomb for the neurons to kill and the neighboring neurons. So this mechanism actually demonstrated that even without the amyloid plaques, we can actually cause Alzheimer's disease by just causing the neuroinflammation. And this critical molecule in this neuroinflammation is this accumulated hydrogen peroxide. Okay, so we can actually redefine the Alzheimer's disease hypothesis that it starts from amyloid plaques, but this presence of amyloid plaques and the presence of the severe reactive astrocyte ensures the onset of this Alzheimer's disease. And this is all through the H2O2, because if we use H2O2 scavenger, we can actually stop all this process. And this leads to tauopathy, brain atrophy, and neuronal death and cognitive decline. And the key component here is the component of the neuroinflammation, which is severe reactive astrocyte, and the molecular factor is this H2O2. And this was published in Nature Neuroscience in 2020. And we also made a movie out of this. So Alzheimer's disease, neurodegenerative disease, meaning the neurons die. And we are the first one to actually demonstrate that the astrocyte kills the neurons. So in the hippocampus, in early Alzheimer's disease, we believe that this amyloid plaques are floating around. Oligomers are also floating around, and this causes these astrocytes to become reactive, start to take up these amyloid plaques and degrade, and that leads to putrescine production. And you can see that the GABA production also produces H2 accumulation. So the difference between the mild reactive astrocyte and severe reactive astrocyte was the presence of this turning on of the INOS, which is an inducible NOS, NOS, I'm sorry, INOS. So INOS was not present in the mild reactive astrocyte, but only present in the severe reactive astrocyte. And as you know, NO is produced. NO can be toxic if it's combined with the superoxide coming from the NOx2. And superoxide and NO can produce peroxynitride, which causes a nitrosative stress and neuronal death. So we saw this severe reactive astrocyte causing atrophy, brain atrophy, which is all recovered, rescued by the this AAD 2004, which is the potent H2 scavenger. So we can actually block this phenomenon by two ways. So we could use MAO-B inhibitor, KDS 2010, as well as this AAD 2004, which is a H2 scavenger. And using these two drugs, we were able to prevent and rescue all this neurodegeneration. And so the story is a happy story. Now we can actually discuss about this major question: why did the amyloid-based drugs fail? And the first question: why AD continues even after A beta elimination using this aducanumab and solanezumab, all these drugs that are supposed to eliminate A beta, why the AD continues? So we could answer this question by saying that even if A beta is eliminated, if there are severe reactive astrocytes, AD can irreversibly continue. Now, most of these antibodies, as you know, causes triggering of neuroinflammation. So because amyloid beta, Alzheimer's disease caused by, if we consider that as a disease caused by neuroinflammation, these antibody-based drugs will exacerbate and enhance the neuroinflammation because it triggers the immune system. So the whole concept, I believe, is not very proper, just to start with. So we need some ways to prevent neuroinflammation, not exacerbate neuroinflammation. The second question is why some people with A beta do not show AD symptoms. It's been reported that even normal population, some normal population has high level of A beta by this PET imaging probes called PIB. People have reported that under various normal healthy controls also show high level, but they don't really show the cognitive impairment. So people have wondered why the A beta is there, but why the symptoms don't exist. And we answer this important question by saying that even if A beta is present, if there's no severe reactive astrocyte or neuroinflammation, severe neuroinflammation or severe reactive astrocyte, there can be no AD symptoms. So we try to explain this in terms of the severe reactive astrocyte. So then lastly, when does A beta accumulation leads to AD? So even if there are normal population with A beta, when does this normal person shows AD symptoms? To test this idea, to address this idea, we have performed a simple experiment. We use the APP-PS1 mouse, which is widely used and popular AD model, but everybody knows that it's not a good model because it doesn't show, this mouse does not show neurodegeneration. It shows the cognitive impairment, but neurodegeneration is not present. So we use this idea, we use this mouse, and we injected a virus, adenovirus, which is known to cause inflammation in the brain, and we injected into hippocampus. And we can see that this APP-PS1 mouse with this virus, we were able to show cause a severe reactive astrocyte. And you can see now that the neuronal population disappears when the APP-PS1 control mouse does not show still shows the neuronal population. So we were able to cause neurodegeneration in this lousy AD model called APP-PS1 because we were able to introduce the severe reactive astrocyte with the virus. So we answered the last question. When does this A beta accumulation leads to AD? I think it's if it accompanies some experience, such as viral infection or traumatic brain injury, which leads to severe reactive astrocyte, can cause such a leads to AD condition. Okay. From these studies, we were able to develop this H22 scavenger, A80 2004, and KDS 2010, which is a MAO-B inhibitor. And these are two compounds are actually drugs are being developed and tested at the clinical level by GNT Pharma and Neurobiogen. So we are actually waiting to hear the result of this clinical tests. We are also in-house developing another H22 scavenger, which is also potent as AED 2004 or better. And this drug also shows a very nice effect of a recovery of the memory. And this drug is also waiting for technical transfer now. Lastly, we have extended our study to ask the question of what comes after the putrescine. Not after, before the upstream of putrescine. And as you remember, I alluded to the presence of a urea cycle. So we actually tested this idea extensively. And to make the story short, it turns out that under normal condition, the astrocytes contain a partial, not a full cycle of a urea cycle, and it's not very robust cycle. But under AD-like condition, when we apply A beta, or in the Alzheimer's disease model mouse, or even AD patients, we see a full cycle of urea cycle, which is fed by the presence of a high level of ammonia, which comes from the degraded products of the amino acids, probably coming from the autophagy and lysosome system. And this leads to degradation of the toxic proteins into various amino acids, including aspartate, and accumulated ammonia feeds into the urea cycle and actually turns on this urea cycle. And when this urea cycle turns on and cycles, there's an accumulation of ornithine, which is one of the major component of the urea cycle. And this accumulation of ornithine, with the help of ODC1 enzyme, produces putrescine. And once putrescine is accumulated, it leads to the GABA production. So this whole pathway has been developed and delineated and was recently published in Cell Metabolism. And we were able to reverse this urea cycle and putrescine production with ODC inhibition. So ODC1 is a very important enzyme that bridges between urea cycle and the putrescine degradation pathway. So if we block the ODC1, we were able to preserve the urea cycle, which is good because it detoxifies the toxic ammonia. It converts the ammonia to urea, which is less toxic, and it gets excreted to urine. And while it's preserving this by blocking ODC1, it blocks the putrescine production and blocks this toxic pathway. So ODC1 can be a very nice target, new target for Alzheimer's disease. And we also made a movie out of this. So the urea cycle was firstly found in this Alzheimer's disease patient brain. And its role is to detoxify ammonia to less toxic urea. However, overload of this urea cycle can lead to overproduction of putrescine, which causes the toxic things like ammonia and H2O2 and GABA produced. This urea cycle is actually well reported in the liver as well as the kidney, but we are the first to demonstrate that this also exists in the brain. And it's very active during the clearance of these toxic proteins. And the normal condition is kind of partial, whereas in the AD-like condition, it's fully functional and cycling. And this causes production of putrescine. So we have actually these two components, urea cycle leading to putrescine pathway. And these enzymes were also found elevated in the Alzheimer's disease patient brain. Urea was also elevated in this AD model or in culture when we treat with the Ibra. And as I said, ODC1 is the bridge between the urea cycle and the putrescine degradation pathway. And this part, urea cycle, appears to be doing a good job. It's very beneficial, whereas this putrescine is detrimental. So the strategy we can use is to block the ODC1 to preserve the beneficial part of the urea cycle and block the detrimental pathway of the putrescine degradation pathway. So by blocking the ODC1, we were able to block the tonic GABA release, and the firing was also rescued. And we found that the behavior was also rescued by ODC1 gene silencing. So ODC1 can be a very effective drug target for Alzheimer's disease, as good as MAO-B. We also found that the treating with the gene silencing shRNA with for ODC1 also reduces the amyloid plaque number. And we found that this is due to the turning on of the non-amyloidogenic pathway. So we can actually achieve the toxic molecule production by ODC1 inhibition, as well as the decrease of the amyloid plaque. So reactive astrocyte has this two phases, dichotomy. One thing is good, and the other is also there, and it's bad. So this dichotomy is explained by our this ODC1 bridging between urea cycle and the putrescine degradation pathway. In addition, we have developed the this PERPEC, which is a 2-photon imaging probe to selectively mark the neuritic type of the amyloid beta plaques, which are known to be more toxic. And this we are trying to develop based on this PERPEC. It's a fluorescent molecule, but we are trying to use that as a PET imaging probe. Also, we are also developing various tools to mark reactive astrocyte so that we can do a early diagnosis of the Alzheimer's disease patients. And we recently found that acetate, which also is a component of urea cycle, it turns on the urea cycle, can be taken up by MCT1, which is overexpressed in this reactive astrocyte, and causes the reactivity and this turning on of urea cycle. So we use this mechanism to mark the reactive astrocyte using the C11 acetate as a PET probe. So that is actually under review now. So compared to 2014, we had the original report, original proposal. Now in year 2020, we almost completed this proposed pathway. And now we are currently working on the autophagy system. And also we are identifying the receptor that causes the uptake of these amyloid plaques in the reactive astrocyte. So with the future projects, ongoing projects, we are working on the autophagy, A beta receptor, and the acetate and TCA cycle, and the PET imaging probe using PERPEC, as well as the MAO-B inhibitor. And we're also doing a drug screening for ODC1. And ASO is the antisense oligomer is also being developed in collaboration with KAIST Professor Jinggu Kim. And so the future of this reactive astrocyte is very bright. We have developed many different tools. Is all this related to pain? Well, I don't know. I mean, somebody has to test it. I think it's highly possible. Under chronic pain condition, it's been reported that reactive astrocytes are there, and people don't know what they're doing. So somebody needs to test it. Well, we have had a small collaboration with Professor Jinbong Park at Chungnam National University. Now he moved to Seoul National University. He tested the effect of KDS 2010 in the paclitaxel induced tactile hypersensitivity model. So this is a paclitaxel is a anti-cancer drug, and this causes some reactivity, and this causes chronic pain. And we were able to show that the MAO-B inhibitor reduces this pain. So definitely there's a strong possibility that these reactive astrocytes are involved. So we are waiting for some collaborators. So this is our center at IBS. I am serving as a director, and we would like to have you invite someday to come and visit us and give us a seminar on your own, and then we can talk more about collaborations. So with that, I'd like to thank you for your attention.
Wow. Professor Tae-Kim, you are muted.
Okay, thanks. Thanks for letting me know. So thank you, Professor Lee. The wonderful lecture and beautiful sciences. I was very impressed with today's talk. So I would like to invite people to ask some questions. So there is one the questioner, Professor Young Kim, who is a visiting professor from MGH.
Yeah, I see the question.
Okay, yeah.
So I can read it. How is A beta triggered to participate in the urea cycle? So that's the first question. And the second question, does the A beta concentration matter? How is the abnormal level of A beta formed initially? Okay, so it's actually a multi-part question. The first question. So A beta can trigger the urea cycle. However, we found that multiple factors are needed for turning on of the urea cycle. So, for example, amyloid beta binding to the receptor. Right now, I cannot identify the receptor yet. But this binding of this receptor turns on the autophagy system. So it causes transcriptional regulation to turn on the autophagy. And autophagy system is highly regulated, highly enhanced. And this causes degradation of the amino acids. So the taken up amyloid beta is degraded. And the aspartate here is needed to turn on. At the same time, accumulation of ammonia, which could come from the this lysosome, is also needed. This ammonia also comes from the MAO-B enzyme. This ammonia accumulation is definitely needed. At the same time, as I said, acetate is needed. Acetate comes and by taken up by astrocyte, reactive astrocyte through the MCT1, and it's converted to acetyl-CoA. And this acetyl-CoA is required for this step where the CPS1 causes the carbamoyl phosphate. So there are multiple factors that synergistically turns on the urea cycle. Okay, the next question is, how is the abnormal level of A beta formed? This is a $1 million question. A beta accumulation is a homeostatically maintained. And I believe the astrocyte is the major cell type that balances the A beta production. And so if anything goes wrong in the astrocyte, such as in severe reactive astrocyte, something is going wrong and it's not doing its job. So severe reactive astrocyte is probably doing its best to degrade, but it's not, I mean, it's the accumulated A beta is accumulated and it's not doing its job. So it goes to this state where irreversible state where there's no turning back. However, how this A beta is accumulated is still unknown. I showed some clue here that this enhanced level of ODC1 is somehow linked to this accumulated amyloid plaque. So if we inhibit ODC1, we were able to actually convert this amyloidogenic pathway to non-amyloidogenic. So I think this accumulation, A beta accumulation, is somehow linked to this urea cycle related enzymes, which regulates this beta secretase, alpha secretase, and gamma secretase balance. Okay, so that's the next question. Functionally, does the A beta concentration matter? Yes, surely it does matter. If it's low enough, it only causes mild reactive astrocyte. If it's continuously present at a high level, it goes to the severe reactive astrocyte. Okay, number 2. Functionally, only GABA is abnormally regulated in AD patients. Only GABA is abnormally regulated in AD patients. Oh, well, many things are aberrantly regulated, not only GABA. As you can see, the ROS production is also abnormally regulated. I think the key is not the GABA itself, but ROS. I mean, ROS is always a bad thing, but although low level of ROS is required, high level of chronic level of ROS can be really bad for the whole environment. So I think it's not the GABA, but it's the ROS, which is abnormally regulated in AD patients. So by blocking the ROS production, we can actually prevent many things. So they are actually interrelated. Okay, third question. Can anyone separate H2O2 induced inflammation from neuronal atrophy? Yes, we were in our 2020 Nature Neuroscience paper, we scavenged H2O2 with AAD 2004, and we were able to reverse or prevent the neuronal atrophy. So definitely we can separate the inflammation, separating the inflammation. Actually, no, because the reactive astrogliosis is caused by H2O2. And H2O2 also causes the neuronal death. So it's actually inseparable. My simple answer is it's inseparable. Okay. If you block the neuronal mechanism, you could separate it, but at the H2O2 level, I don't think you can. I mean, whether the neuronal dysfunction alone could cause the structural atrophy. Well, this approach to look at the neuronal dysfunction alone is the traditional way to look at the Alzheimer's disease. And as I said in the introduction, that approach has failed, continuously failed. And I think just looking at the neuronal dysfunction is not a good approach to address the Alzheimer's disease. So I think we should look at the whole brain as a whole, and the astrocytes that are nearby the neurons, which is supposed to support the neuronal function, when they go wrong in a condition like Alzheimer's disease, everything can go wrong, and even the neuronal death can occur. So I think focusing on the neuroinflammation and reactive astrocytes, we'll find the answer much quicker, and we'll find a better answer. Okay, so that was a long list of questions.
Yeah, thank you very much. Yeah, nice question and answers. But I think that there should be more questions from other participants. So if you have, you can turn on your microphone and you can say, or you can leave a question in the chatting window. So meanwhile, I can add one more question in relationship with the question number one. So after A beta concentration matters, as you answered. So aducanumab, actually it can reduce the A beta concentration, but it still continues the dementia progression. So that is a problem. But actually, based on your theory, the A beta is reduced, then the source of putrescine is also reduced, and all other pathological process will be also reduced. Then why the severe reactive state could exist or continue?
Yes, so that's a very important question. So this is what I believe, and I mean, I don't have direct evidence yet, but it's been very well known that aducanumab, as well as other antibody-based drugs, causes ARIA, which is actually neuroinflammation. So this antibody treatment can cause inflammation. So instead of inhibiting inflammation by removing amyloid beta, it actually exacerbates neuroinflammation. So either the people have to design an antibody treatment to remove amyloid beta that does not cause inflammation, or they should drop all this this approach. This is pointless, I think.
Okay.
As long as this antibody treatment causes ARIA or further enhancement of neuroinflammation, this won't work. I guarantee you, this won't succeed.
Okay, so
You need to block the
that
inflammation.
is due to it is an immune mediated
That's
response.
right, yeah.
So immune is more activated so that now the reactive astrocytosis is exacerbated or continues.
Yeah. Perhaps if we could combine this aducanumab with like KDS 2010, which inhibits inflammation, then it could work.
Right, right.
But I mean, the companies have to buy this idea and test it. I mean, we can.
Or ODC1 can also combine as well to reduce the neurodegeneration.
That's right. Yes.
Okay, so there's one more question from in the chatting window. Can you see that?
Yeah. So question, it sounds neuroinflammation is important. How does the astrocyte play a role for various diseases, including AD, Parkinson, etc.? Can this be the key treatment for those diseases? Answer is yes. We have tested the same drug in Parkinson disease model, and it's very effective. In fact, the MAO-B inhibitors has been used in Parkinson disease. However, as I said, the people have used irreversible inhibitors like cellular and razajulen, and these irreversible inhibitors only has a time window of kind of short period of time. So it's been known that MAO-B inhibitors are useful for a short time, but it's no longer useful. So using these reversible inhibitors, I think it'll be very useful for Parkinson disease. And yes, for other diseases that shows strong neuroinflammation, I think these drugs can be very useful. And if chronic pain is related to inflammation, neuroinflammation in the spinal cord or in other brain areas, I would definitely, I would almost guarantee you that it'll have a very good effect.
Yeah, I think that sounds too promising. So, for example, as you say, Parkinson's disease. So, like, you mean that the reversible MAO-B inhibitor, like KDS 2010, has been shown to treat Parkinson's disease? Is that like, because I don't know much about this?
Yes, in fact, there's a competitive drug called safinamide, which was recently FDA approved, I think last year or a couple of years ago. And this is used for Parkinson disease. So, and this is also reversible inhibitor. And I'm hoping that this reversible inhibitor will show much better efficacy compared to the irreversible inhibitor, the traditional cellular and razajulen.
Okay. Yeah, I guess regarding to that, I have maybe connecting question.
For applying the MAO-B inhibitor to the pain patients, how to deliver it through the BBB and only intrathecal administration works? No, actually this MAO-B inhibitor is highly BBB permeable. And most of our experiments with mouse is by drinking. Adding it to the drinking water. So it's very easy to administer, and you don't really need to use intrathecal injection.
Okay, I see. That's very promising. So another maybe I don't know whether there's a lot of content you describe linked to neuroinflammation, and it's not only about AD, you have shown various very important neurodegenerative diseases. So I wonder if is neuroinflammation as a result of these different pathology, or neuroinflammation causes these various neurodegenerative diseases. Depending on this,
Yes,
I guess the treatment strategy might be different.
right. So in the beginning of my talk, I tried to explain to you that most of these drugs fail because people have ignored this neuroinflammation, believing that it's a byproduct of neuronal death or some kind of toxic molecule. Presence of toxic stuff causes this reactive astrogliosis. So people have tried to consider that as a byproduct of the disease state. What I'm trying to say is that you cannot ignore this event because that is the beginning of the disease. So in the beginning, when the brain or these glial cells surrounding the neurons are enough or good enough to take care of the problems, then the brain will be fully back to normal after the initial challenge, toxic molecule challenge, and then astrocytes and microglia are enough to cover, eliminate all the toxic stuff, and so the astrocytes become mildly reactive, and then they recover, and after that is fine. There's no problem. The problem only occurs when this toxic stuff prolongs and lasts a long time and chronically present in the brain. So the astrocyte, initially starts to eliminate this, clear this out. But then if it lasts long and long enough, that's when this symptoms can occur. So initially when there's accumulation of some toxic molecule, including amyloid beta, it's no problem because astrocytes and microglia, if they can take care of it, no problem. There's no further progression of the disease state. However, if the load of the challenge or the toxic stuff surpasses the capacity of the astrocyte and microglia to clear all this out, then the disease symptoms appear, I think. So in a way, the brain inflammation is the cause of the disease.
Oh, okay.
So, I have a simple question that the
It's
A beta.
small, easy example is the normal population with high level of A beta, but they don't show the symptoms. Because I think astrocytes and microglia are doing its best to clear it out so that it doesn't really cause severe inflammation.
Oh, okay.
Yeah, I think it can be a very good explanation about the preclinical AD patients who has the quite high level of the amyloid in the amyloid PET, but they don't have any symptom of dementia. So that is almost 30% of amyloid PET positive patients. So that is actually the question to the Alzheimer's disease researchers, and this is a very good explanation about that. I have one simple question that you apply this theory into the Parkinson's disease, then can I understand that the A beta site, the only replace it with the alpha-synuclein, then it is all the processes same?
Yes. Alpha-synuclein can be the similar cause of this accumulation of the toxic molecule. However, in the substance at Niagara, there's another strong component, which is the ROS So
Mm.
the whole area, substantia nigra, is vulnerable to ROS because dopamine itself is highly can produce lots of ROS by itself. So it's the area where this can occur more prevalently, more often than Alzheimer's disease. I think it's actually substantia nigra is more susceptible to a challenge like this
Okay.
because of
So,
the presence of the ROS stress.
maybe that's why the only the dopaminergic neuron is more affected in alpha-synucleinopathy.
Right.
Hey, thanks. Is there so the time we have
There's one more question,
we
I
have
think.
more time, then oh yeah, this was not the
Okay.
question. Professor Hwang Jae-yoon, due to the time, he left earlier. Okay, so maybe we have to wrap up today's the colloquium seminar, and I think it was
I have
really
one more question.
okay, last one.
Sorry,
Of course,
Please.
sure.
Sure.
Sorry for the interruption. Mostly, I think your talk is focused on this astrocytes and reactive astrocytes, astrogliosis. And if we consider about other glial cells, and I wonder if there are like microglia and
Right,
also oligodendrocytes or ependymal cells. Some different types of glial cells are existing inside the brain. So I wonder if is the astrocyte is shown to be very important. But what about other like major player for maybe neuroinflammation, like microglial role in AD or Parkinson or other neurodegenerative disease? Maybe
So
comment on that.
this is a very critical question. I do believe that microglia is as important as astrocyte. I didn't show today, but we've been collaborating with Professor Jo Han-sang at Sungkyunkwan University. And the question that we asked was that which one is more important? Is it reactive astrocyte or is it reactive microglia? And bottom line was that it appears that reactive astrocyte is the first to appear, and then it triggers or recruits the microglia to the site of injury or site of the toxic stuff. And this microglia invasion to the site of injury causes or exacerbates or boosts the toxic effect. So like with astrocyte alone, it might take I don't know 2 days to kill neurons, but with the help of microglia or toxic microglia, it'll take less than a day or so to kill the neurons. So they actually work together. They call each other, and they work together to make it worse or exaggerate this inflammatory response and toxic effect. And but reactive astrocyte seems to be enough alone without microglia. It can alone kill the neurons, and it also invites the toxic microglia to come to the site of injury. So that's it's not published yet, but it's in the pipeline to. I think it's submit, and it will be coming out soon.
Okay.
Oh, that's very interesting. Thank you. It sounds like revenge of these glial cells to kill the neuron.
Revenge. I would say, I mean, they're a good thing. The astrocyte and microglia, they are doing its best to maintain the brain, but they became somehow killer. So we need to figure out why it's happening, and then we can stop this process.
So to kill the enemy, they use two strong weapon, and
Yeah.
then they kill themselves together.
Well, they don't get killed. I mean, these reactive
I mean,
astrocytes are very strong.
they're our own
Ah,
cell.
yeah, that's the neurons.
Yeah,
Shouldn't
neurons.
be
Yeah.
killed, but they are killed
Right,
by this
right.
process, which
Mhm.
is unfortunate story.
Right.
So
Some
we
kind
need to
of
stop this.
collateral damage.
That's right. Yeah.
Yeah. Okay. Thank you so much. And today's talk was I think it was one of the most relevant and useful the seminar, because our group is a gliopathic the GPRC, the gliopathic research.
Oh, that's right. Yeah, I think that G stand. I thought
Yeah.
standed for gist, but it was actually glia.
Yeah. So it was very fortunate for us to learn about the most advanced and the recent cutting edge knowledge. So thank you so much about this talk.
Thank you very much for invitation.